You can start, sir.
Uh, good evening, everyone. This is Dr. Abhijeet, your biochemistry educator at Auricle. And today, in this Oracle 90 Days Foundational Life Class for University Exams, the topic that we are going to cover and discuss today, it is the proteins and amino acids biochemistry. Guys, today I will be revising the important questions related with the protein chemistry, and it will be a second discussion. The first we have already discussed the previous week. So I will be revising all the potent questions which I feel that will be like important for your upcoming examination. Right. Even I will be covering all the previous 10-year questions as well that have been asked in the various university exams, various prof exams across the globe, right? And guys, it is like beneficial for everyone who's gonna appear for the university prof exams and the next exam, right? Now, the questions that I will be covering, it is divided into three sections, guys. We have the long essay section, which consists of 10 marks question, the short essays that are 5 or 8 marks, and the short answers that are 2 or 3 marks. In totality, I will be covering about like 7 questions today. And with each question, I will be discussing each topic individually, right? Now, guys, you already know about the Oracle app, right? So let me just go through it. We have the recorded classes, guys. Once you are not able to see the live sessions, you can just go on for the recorded versions which are available on the app, which is available on the app. We have the live classes that is being scheduled daily from 6 to 10 p.m. and all the educators at Oracle, they are like teaching. Okay. We have the practical sessions which are recorded also, and which I will be also teaching from for my subjects like later on after the theory completion. We have the express exam module, right? See, just 1 month before the exam, I will not ask you to go through the textbooks. Yes, just some days left, and you just want to revise the entire biochemistry. That is this exam module. It is basically covering all the essential topics in under 20 hours. And apart from that, guys, we have the hard copy book set that you can just order, and it is curated in the form of question and answer format. That is the same way, guys. Like you have to write the answers in your exam as well, right? Now, the recorded classes we have already discussed about this, right? So flexible learning. Whenever you have free time, you just can go on for the recorded versions. We have the live classes that we are conducting daily from 6 to 10 p.m. Right? We have the one-to-one doubt clearing session as well, right? You can anytime book a slot and clear all your doubts related with the topic and the concept that you are studying. And apart from that, the best part is that these hard copy book sets like you can order, it's like available doorstep, right? So they are covering all the important questions in the form of Q&A, all the puzzles, previous 10-year questions, all everything is there in these textbooks. We have the question bank that is from basic to advanced level, guys, right? So we have like covering the questions from like past 10 year, whether it is any INI-CET exam or NEET PG exam or FMG exam or university prof exams. MCQs are also there. And the advanced level questions, guys, nowadays the pattern is more clinical, right? So we have clinical vignettes, and that is being created by our expert team, and that are two there in this particular Q bank. So you can just, and after every like question there is like explanation of that particular topic as well. So yes, we have the practical sessions which are being recorded, and that will be like a helpful aid just before the practical exam so that you can excel well in that case. So we have the basic subscription plan, the foundational plan, that is yes, you can just go on for this for 3 months, for 6 months, for 12 months, and for 4 years, right? For additional discounts, guys, you can just use my code Dr. Abhijeet 20 for discount on this particular subscription, right? Now, coming on to the very first question, that is of 10 marks. So, all the important points you have to just write. So, the question says, describe the structure and levels of organization of proteins, primary, secondary, tertiary, and quaternary structure. Discuss examples of each level and their importance in protein function. So we are going to discuss the various levels of organization of proteins, the primary, secondary, tertiary, and quaternary structures. So basically, the structure of proteins, it is basically divided into four levels of organization: primary structure, secondary structure, tertiary structure, and quaternary structure. Now, guys, you already know, whenever let us just compare with the English alphabets, guys. You know, we have the English alphabets, right? We have A, K, C, D, M, N. So just like we have primary level, then you what do you do with these alphabets? You make words, right? Then basically, you arrange them and you make words. From words, we have sentences, tertiary structure, and from sentences, we have like paragraphs. So similarly, in this case also, guys, we have like four levels and they are just based or comparable to this particular. So we have amino acids and like the primary structure, then secondary, then tertiary, and then quaternary structures. Just like the English alphabets arranged, then it is forming words, then meaningful sentences, then meaningful paragraphs, right? So let us just start with the primary structure, guys. Primary structure. Now, what is this primary structure of protein, guys? Let us just try to enumerate something over here. So guys, remember the primary structure of the protein, it is a linear. Now guys, very, very important. It is a linear sequence of amino acids. We have discussed amino acids in the previous part of the protein chemistry, right? We have discussed amino acids, right? We have 20 amino acids, right? We have discussed essential amino acids, non-essential amino acids, right? We have discussed the amino acids based on the variable chain, right? Based on the ketogenic, glucogenic fate, nutritional requirement, essential and non-essential, semi-essential also. So we have like 20 amino acids. Now it says that obviously whenever amino acids will be joining with each other, okay, and that too in a linear sequence, guys, that will lead to the primary structure of the protein. So it is the linear sequence of amino acids. And guys, we already know that two amino acids are joined together by peptide bond. They are joined together by the peptide bond. And peptide bond is basically formed from the loss of a water molecule, right? So this peptide bond is nothing but C double bond O NH, right? So this is the peptide bond. This is the peptide bond. This is the peptide bond. We have already discussed whenever two amino acid will join with each other, there will be a removal of a water molecule, right? We have already seen in the previous section. And after removal of the water molecule, what will be there? There will be C double bond O and NH. Okay. You already know one amino acid is having COH group also and NH2 group also. One H and one R. This is one amino acid. Another amino acid will be also having this C double bond O. Okay. This NH2, this R and this H. So yes, we have two amino acids. And if there is removal of this water molecule, then what will be remaining? This CO and this NH. So this is something called as the peptide bond. So this is the primary structure of the protein, guys. This is the primary structure of the protein, right? So yes, the primary has a unique sequence of amino acid, which is determined by the genes contained in the DNA, right? Now, the primary structure, okay, guys, let me just take an example over here. Yes, let me just take an example so that it is like way clear to you as well. So if we take an example over here that is called as insulin. Now, insulin, guys, you already know that the insulin, it is basically secreted from which cells of the pancreas? Yes, from the beta cells of the pancreas. Okay, alpha cells secrete glucagon. So we have beta cells of the pancreas which secrete the insulin, right? Now you already know insulin is a hormone in the body. It is a hormone in the body which regulates the blood sugar. Okay, which regulates blood sugar, right? Which regulates the blood sugar. Now this insulin, guys, this hormone, it is nothing but, guys, remember that it is consisting of a specific sequence of amino acids, right? So it contains sequence of amino acids. Okay. So it contains sequence of amino acid. Just a moment. So it consists of sequence of amino acid, and guys, you already know from the physiology part, it consists of 51 amino acids. Very, very important question from the exam point of view as well. Okay, it consists of 51 amino acids, and remember, all these 51 amino acids will combine with each other in a linear structure, and it will form the hormone that is called as insulin, which is regulating the blood sugar in our body, right? Which is regulating the blood sugar in our body. So now you already know the amino acids are linked together by protein by covalent peptide bonds. Covalent means beta by sharing. Obviously, by sharing of the electrons, this water molecule is removed, then this bond is formed, right? C double bond O N H. Okay. So obviously peptide bond. Why? Peptide means protein only, guys. Peptide means protein only, obviously, right? And obviously, whenever there is sharing of electrons, these bonds are very, very, very strong. These bonds are very, very strong, right? Now, how this peptide bond is formed, we have already discussed it. If you want to like know over here, let us just try to see over here, guys. Let us just try to see one example. Okay, how this peptide bond is formed. Now, guys, let me take an amino acid. Okay, let me take an amino acid. Already I have told this, but just to make it more clear, guys, let me just take it. Okay. Now you guys already know we have a carbon, and this carbon, it is containing COOH group. It is containing NH2 group, that is called as ammonia. It is containing one R group. R basically refers to the number of carbon atom. R refers to the number of carbon atom. It can be like 1, 2, 3. So carbon and hydrogen, right? And there is one hydrogen over here. So this is the basic structure of amino acid, right? Now, one amino acid, it is combining with another amino acid. And you guys already know we have carbon. This carbon will be having the COH group, will be having this ammonia group NH2, will be having this R group, and will be having this H group. So we have two amino acids. This R can vary. That is like the various 20 amino acids are there, right? Depending upon the number of carbon atoms. Now, what is the funda over here, guys? You just have to remember that the water molecule is lost. OH, water ka formula. What is the formula for water, guys? You already know H2O, right? So this OH from the carboxylic acid, and okay, and this one hydrogen from this, yes, NH2. Now what will happen? Again, the same thing, guys. This carbon, this carbon will be down. Okay. Now what is left? CO is left. So it will be C double bond O, I can write, right? CO means C double bond O, right? I can write like this, C double bond O. And this one hydrogen is removed. So we have NH only remaining. It is not NH2 now. One hydrogen is removed. So what will happen? There will be one nitrogen and one hydrogen. And again, the other structure, it is like same, right? The other structure, it is same. So this bond, guys, this bond, let me just correct it. Yeah. This bond, C double bond ONH, it is the peptide covalent bond. It is the peptide bond, right? So this is the peptide linkage. This is the peptide linkage. Okay. This is the peptide linkage, guys, right? So that is something you guys have to remember from this particular part. So formation of a peptide bond when the amino group of an amino group means NH2 group of an amino acid combines with the carboxylic group of another amino acid, there will be result in the peptide bond. Dipeptide will have two amino acid. Obviously, guys, just see Whatever we have formed just right now, it is a dipeptide. It will be having two amino acids. One amino acid is this, another amino acid is this. So the dipeptide will have two amino acids and one peptide bond. So obviously, you can just see from over here also that it is having two amino acids, right? It is having two amino acids and there is only one peptide bond. Only one peptide bond, right? So yes, only one peptide bond. The peptide bond is rigid, planar, with partial double bond in the character, right? Obviously, it is like formed by the sharing of the electrons only, right? So that is the primary structure of the protein that you have to remember. That is the primary structure of the protein you have to remember. Now, guys, what is the importance over here? What is the like if I just want to discuss the importance also over here. Now, guys, just try to remember what is the importance. Because the question also says we have to discuss the, yes, example. So I have seen, example I have just told you, insulin, okay? And the importance in the protein function, okay? Now, guys, what is the importance of this primary structure? Obviously, guys, if we are taking certain words, A, K, C, L, O, these are words. Now, these words have to be arranged in a specific order. Okay, these alphabets have to be arranged in a specific order to form a meaningful word, right? So the primary structure actually tells how the protein will fold, okay, how the protein will fold, okay, and consequently it will give the shape of the protein, okay. Even a single change in the amino acid, guys, even a very, very small change in the amino acid, it can lead to major diseases. For example, we have discussed point mutations in genetics, right? Insertion, deletion. For example, like you already know sickle cell anemia. So if there is any like change in the single amino acid also, the entire change will change. I mean, the entire protein will be disrupted. The primary structure will be disrupted, and it will lead to some kind of disorder, right? For example, sickle cell anemia, right? So that is something, guys, you have to remember from this particular part. That is the primary structure of the protein. That is the primary structure. So we are done with the primary structure of the protein with example and importance, right? Now coming on to the next one, that is the secondary structure of the protein. The secondary structure of the protein. Okay. Now, we have a primary structure. Now, when I say primary structure, guys, it is always the linear structure, just like a straight line. Secondary means it is this linear structure, it is getting folded. It is getting folded. So basically, the secondary structure, it is called as the folded structure. Okay. So this is basically, okay, let me just write down that it refers to the localized folding patterns. Localized folding patterns, okay, within the protein, guys, within the protein, okay, and they are stabilized by a bond that is called as the hydrogen bonds. Very, very, very important. Okay, they are stabilized. Okay, now can you see the structure, guys? It is like folded structure. It is not a linear, it is getting folded, right? Now, in these folded structure, in between the foldings, guys, we have these hydrogen bonds, which are basically responsible for the stability of the secondary structure. Okay. For example, let us start with the alpha helix. Alpha helix, Peter, remember it is a right-handed coil. Okay, it is a right-handed coil. where each turn of the helix, if you can just see the diagram also, where each turn of the helix, okay, contains about 3.6 amino acid. Okay, it contains about 3.6 amino acid. Okay, now, now guys, you already know that it is the most common spiral structure of a protein. It is a rigid arrangement polypeptide. Polypeptide beta nothing. It is actually the protein only. It is actually the protein only, right? So alpha helix, it is a tightly packed coiled structure with amino acid side chains extending outwards from the central axis, right? Now, over here in this structure, guys, in this structure, the amino acids, they are tightly packed with each other. Okay, they are tightly packed with each other. They are basically coiled, and where the side chains. Now, when I say side chains, guys, I'm referring to the R group. These are basically the carbon and the hydrogen group, the number of the carbon atoms, they are extending outwards. Suppose this is the central axis, they are extending outwards from the central axis, right? So alpha helix, it is like stabilized by the hydrogen bonding. Now this hydrogen bond, guys, it is formed between the hydrogen atom attached to the peptide N, okay, and oxygen atom attached to the peptide C. So we have like two terminals, guys, the N and the C term, right? So guys, remember as of now that these hydrogen bonds are very weak. Okay, they are very weak, but okay. Now suppose if I'm a very weak person and I have like 10 persons like this, weak. And if we combine all these 10 persons, so collectively, guys, we can stabilize the helix. We can like so that is something like going over here also. The hydrogen bonds, if we are just talking about a very single portion, it's very big. But collectively, guys, it is producing a very strong effect. Okay, and that strong effect is no essential to stabilize the helix also, right? So yes, I have already told you each turn of a helix contains 3.6 amino acids. It's very important. Again, a very important question from the exam point of view. And it travels a distance of zero point 0.4 nanometer, 10 raised to the power minus 9, right? And the spacing of each amino acid is 0.5 nanometer. These are the mathematical terms, guys, that you have to just remember. So yes, coming on to the beta 1, guys. Beta 1, basically it is composed of strands that are running alongside each other, and they're connected by the hydrogen bonds, right? So so we have the like parallel and the anti-parallel sheets, guys. You can just see over here, we have a beta strand, right? We have a beta strand just like the pleats of like the skirt, which you can see the pleats of a sari, right? Similarly, the beta strand, which you can see, it is a flat arrow pointing towards the carboxylic end, the COH end, okay? And obviously the hydrogen bonds are basically formed in between these pleats, right? In between these pleats, guys, right? So now, guys, if we talk about the example, if we talk about the example, for example, you already know we have keratin. Keratin, it is basically found in here, and it is also found in the nails. It is also found in the nails, right? Now, they contain a significant amount of alpha helices, right? Okay. Or if we want example of beta 1 also, we have like silk fibroin. Basically, they are made up of these beta sheets, these beta pillated sheets, right? So that is something you have to remember. Now, what is the importance of the secondary structure? Importance of the secondary structure. Remember, it contributes to the protein's stability, and it defines its overall shape. Very important. It defines the overall shape, which is no essential, which is very essential for interaction with other molecules. important for interaction with other molecules. Interaction with other molecules, which is very, very important for interaction with other molecules, right? Now, coming on to the tertiary structure of a protein, right? Now, when I say tertiary structure, guys, remember it is a three-dimensional or a 3D structure, guys. It is a three-dimensional shape, right? It is a three-dimensional structure of a single protein molecule, okay, of a single protein molecule. Now, this three-dimensional structure, it is basically formed by, it is formed by these secondary structures only, right? Right? Formed by the secondary structures, and they are stabilized. Continuous foldings, right? Foldings, you can just see in the images, foldings of these structures, right, will again lead to these tertiary structures, right? So we have various bonds that are participating. We have hydrogen bonds that we have already discussed in the secondary structure. The hydrogen bonds, although they are weak, but whenever they are like having a combined effect, it is making or it is stabilizing the protein structure. Okay, in the starting, we have the peptide bond, we have the hydrogen bond in the secondary structure. In the tertiary structure, we have these hydrogen bonds also, right? We have the hydrogen bonds. Okay. Now, we also have these sulfide bonds in between. If you can just see over here, guys, in this particular structure, we have these disulfide bridges. Disulfide bridges. Okay. Along with that, we have the ionic bonds as well. Ionic bonds. And we also have the hydrophobic interactions. Hydrophobic interactions. So that is something you have to remember. So remember, guys, it is a three-dimensional arrangement of a protein. Okay, it is a compact structure with hydrophobic side chains. Okay, hydrophobic part, the one which is water heating, it is like held interior, while the water loving part, it is held outside. Okay, so we in the interior part we have the hydrophobic, and on the outer part we have the philic, the water loving part. Okay, now this type of arrangement ensures the stability also, obviously. So bonds, basically hydrogen bonds will be there. We have disulfide bonds, ionic bonds, right, and hydrophobic interactions, right? These hydrophobic interactions which are inside, they will be like combining with everyone, and this is nothing which is collectively called as the hydrophobic interactions, and together all these bonds will be contributing to the tertiary structure of the proteins. Now, guys, in the tertiary structure, you guys have to remember one of example. Okay. Okay. Okay, let me just tell you an example that is called as the myoglobin. That is called as myoglobin. Now, this myoglobin, it is basically a protein. It is a protein that carries oxygen in muscle cell. It carries oxygen in muscle cells. Okay. It has a compact globular structure. Compact globular structure. Okay. And this is something which helps to bind the oxygen effectively, right? It helps to bind the oxygen effectively. Now, this tertiary structure, what is the importance, guys? What is the importance of the tertiary structure? It is very important, guys, for the protein's function. Okay, for the protein's function. For example, we have taken the example of myoglobin, right? For for a protein to function normally, or for a protein to function also, this is very important, right? Now, guys, remember, basically this kind of a structure actually determines the active sites. We have some active sites, okay, and on those active sites there will be binding sites also, so that like there could be an interaction, okay, with other molecules or substrates or something like that. So this kind of structure basically like provides some active sites where something can come and bind, just like a receptor. Receptor is also a protein. Okay, a hormone will come and bind to the receptor, lock and key mechanism, just same like that. So this kind of structure basically ensures some active sites where like something can come and bind, and automatically it will result in the function of the protein. That is like way important. Now, coming on to the quaternary structure of the protein. So quaternary structure basically involves there is an accumulation of multiple polypeptide chains. Okay, multiple polypeptide chains. These are the subunits. Sorry, health is not so good. Yeah. Multiple polypeptide chains or the subunits basically into a larger protein. Okay, into a larger protein complex, functional protein complex, basically. Okay, so basically they contain two or more polypeptides which can be identical or they can be unrelated as well, right? So such proteins are termed as oligomers. Oligo basically means two or three, okay, and they kind of like possess this quaternary structure. Okay, a dimer consists, now when I say dimer, di means two guys, di means two. It consists of two polypeptides. It consists of two polypeptides, while a tetramer has four bonds in a quaternary structure, right? So remember, guys, the example over here, we have the hemoglobin, right? We have the hemoglobin which transports oxygen in the blood. So this hemoglobin transports oxygen in the blood. Okay, it consists of four subunits, guys. It consists of four subunits. Okay, we will be discussing this in detail when I will be covering the topic of hemoglobin. It consists of two alpha subunits and two beta subunits. It consists of two alpha subunits and two beta subunits, right? It consists of two alpha subunits and two beta subunits. Okay, and they basically combine or work together. Okay, they work together basically to cooperatively bind to the oxygen, cooperatively bind to the oxygen, right? Now, when I say the tertiary structure is for the like function, for the function part, the quaternary structure, the importance is in the regulatory mechanism, in the regulatory mechanism. Okay. For example, we have taken the hemoglobin that is given in the notes. Now, okay. Now, you already know. For example, if the oxygen is binding to one site, okay, or binding of oxygen to one subunit. So we have four subunits in the structure of this hemoglobin. If the oxygen is binding to one subunit, okay, what it will do, it will increase the affinity of the other subunits also. Like we have four subunits, two alpha, two beta. If oxygen is binding to one subunit, it will automatically increase the affinity of other subunits so that more oxygen can bind over here, more oxygen can bind over here. So this is not only functioning, but it is regulating the work of the entire molecule, right? So that is this. What is the importance of this quaternary structure? It is basically involved in the regulation of these things, right? So, and if if it is okay, oxygen is coming and binding to only one, that will not give an efficient oxygen transport. To give an efficient oxygen transport, this has to bind to the entire or all the molecules. So that is again very, very important, right? So that was regarding the very first question that we have discussed today. That is the primary structure of the proteins, secondary, tertiary, and quaternary. We have discussed the examples and we have discussed the importance, right? We have discussed the importance, functional, regulatory mechanism, right? So that is you all the important points along with the basic diagrams and interactions you have to mention, right? So this is the hemoglobin structure, right? So yes. Coming on to the next question that I have already discussed in the first part, the alpha helix and the beta sheet. and their role in protein stability. Now, guys, you already know the alpha helix. It is a right-handed coil, right? It is a right-handed coil where each turn of the helix contains about how many? 3.6 amino acids. It is stabilized by hydrogen bonds, okay? And it creates, guys, if in words you just have to remember, it creates a compact cylindrical shape. It contains a compact cylindrical shape kind of a structure. Compact cylindrical shape. Now, what is the role in protein stability, guys? If we talk about the alpha helix, what is the role in protein stability? Role in protein stability. Now, guys, you already know we have hydrogen bonding. Now, there is extensive hydrogen bonding in this alpha helix structure, which no, contributes to the stability. Along again, guys, I'm telling you the hydrogen bonds, if they are present single, they are very weak. But obviously, whenever there are like multiple hydrogen bonds, collectively they will produce a very stronger effect, and that is basically making the structure like resistant to denaturation. Resistant to denaturation. Denaturation basically means yeah, the degradation of protein structure. So this hydrogen bonding, it is basically protecting the proteins to get denatured. And that is how it is helping in the stability of the protein, right? Apart from that, we also have the compact structure, guys. We also have the compact structure. Now, if I say the helical shape, guys, the helical shape, the cylindrical shape. So this helical shape, what it will do, it will allow the protein to like pack very, very like close. Okay. The proteins are packed closely, right? The proteins are packed closely. So it is minimizing the hydrophobic part or the hydrophobic residues to the aqueous environments. So obviously it या ये म्यूट कर दो सुनीला प्लीज म्यूट योरसेल्फ hmm. Now we have certain called as the functional sites. Now arrangement of amino acids on the helix. Okay, we have this helix and we have like amino acids. So this arrangement, okay, can create specific. Okay, it can create specific functional sites. Okay, so that can no be a part where no like other molecules can come and bind, and which is very very essential for this biological activity. Coming on to the beta part, guys. Beta part, it is basically formed by strands of amino acid. It is formed by strands of amino acids, right, running along each side. Okay, they are connected to the hydrogen bonds in between. Okay. So now, strands basically can run in two directions, guys. Either parallel, both running in the same direction, or anti-parallel, where one strands run in the opposite direction, right? Anti-parallel beta sheets, like they're very, very like more stable, guys. They are more stable due to optimal hydrogen bonding patterns. There's more hydrogen bonding patterns over here, but they are like very, very stable, right? Now, this also like helps in to withstand the denaturation of the proteins. Obviously, the hydrogen bonds, they provide substantial stability, right? Apart from that, in the beta sheet, guys, remember we have like extended flat surfaces. Now, these flat surfaces can create large areas, okay, for protein-protein interaction. Protein and protein can interact with each other, which can lead to the formation of protein complex, okay, which can lead to the formation of protein complex. And obviously, it can function well, right? So that is something you have to remember. Coming on to the next question, guys. Discuss the protein denaturation, causes, examples, and clinical significance. Now, protein denaturation, guys, you just have to remember it is the process by which proteins lose their native structure, whether it is primary, secondary, tertiary, or quaternary. Proteins lose their native structure, okay, due to the disruption in the stabilizing bonds like hydrogen bonds, or if there is any disruption, right? Or like the bonds are the ones which are no actually maintaining their shape. If the bonds are disrupted, obviously it will lead to the denature of these proteins. Okay. So if the structure is denatured, obviously there will be a loss of function as well. There will be a loss of function. So that is something referred to as the denaturation of the proteins. Now, what are the various factors, guys? What are the causes, guys? In a nutshell, if you just have to remember the heat, the pH change, the the chemical agents, the mechanical forces, the mechanical forces. Okay, mechanical forces just like the physical agitation, right? Such as shaking, stirring. Okay, so that can also lead to the chemical agents, especially urea, guanidine hydrochloride, or detergents can disrupt non-covalent interactions, and that is something which can lead to the denaturation. Okay, for example, guys, this urea, okay, this urea we are actually using in our laboratory settings to now unfold the protein structure for analysis. pH changes can also affect the right the charge of the amino acids, right? So, for example, like you already know enzymes. Enzymes work on optimal pH. So obviously, any deviation in the pH can lead to it in the denaturation. Even the heat can disrupt the hydrogen bonds, it can disrupt the ionic bonds, it can disrupt the hydrophobic interactions. For example, cooking eggs, okay, causes the proteins in the egg white to or denature. Just it like forms a transparent liquid to solid white. So that is how you just have to remember. So even in the notes they have given about this. So yes, yeah, the process of disorganization of native protein structure is called denaturation. So it involves the loss of secondary, tertiary, quaternary structures without breaking the primary structures. Denaturation causes the loss of 3D form of the structures, right? So yes, physical agents and chemical agents we have already talked about: physical pressure, heat, X-ray, UV radiation, and ultrasound. The chemical agents, acid, alkali, organic solvents, either ether, alcohol, or urea. I already told you that helps to unwind the structures, right? So that is you have to remember. Now, what are the characteristics? So the native helical structure of the protein is lost. I already told you the primary structure is against like maintained, but the native structure of the helical, the alpha helix and the beta sheet is lost, right? The primary structure remains intact. Denatured protein loses its biological activity. Obviously, it will not be able to function. Okay, it is insoluble in solvent, right? For example, water, it is insoluble. Okay, otherwise it was originally soluble. So denaturation increases the presence of ionizable and self-hydryl groups due to loss of hydrogen and disulfide, right? So denaturation is typically irreversible, guys. Remember this. Okay. And the denatured protein cannot be crystallized. Now, guys, this is something which we have already discussed, right? Describe the different forces stabilizing the protein conformation, right? So we have already like discussed all these, the hydrophobic interaction, the ionic bonds, right? Now, when I say hydrophobic interaction, guys, let me just write you in short. The hydrophobic interactions occur when the non-polar, or I can simply say the amino acids, the hydrophobic amino acid side chains aggregate to avoid contact with an aqueous environment. That is the hydrophobic interactions, right? Obviously, guys, when the proteins fold, okay, what will happen? This hydrophobic residues, they basically cluster together. Okay, they are coming together in the interior of the protein. I've just told you they are coming in the interior of the protein because outside is water. So phobia means they will not come in contact with the water. So they try to protect from the surrounding water. And this clustering actually minimizes the protein's overall free energy. And just we have a more stable structure, right? Now, hydrophobic interaction, guys, remember they are very, very important for like maintaining compact. Okay, let me just write down. Maintaining compact globular shape of proteins. Globular shape of proteins. Okay. Significantly influencing their folding stability. We have ionic bonds. Basically, we ionic bonds, you already know we have positively charged and negatively charged side of amino acid. Positively, basically the basic, and the negative, the acidic side chains of amino acid. For example, like you already know, we have arginine. Arginine is positively charged, right? So when arginine basically interacts with glutamate, it is again a negatively charged amino acid, and basically they are like interacting with electrostatic interaction. Obviously, positive and negative, we have which kind of interaction? Electrostatic interactions are there. So these interactions are very, very sensitive to pH as well, the changes in the pH and the ionic strength. Okay, so this also like stabilize the protein. Okay, coming on to the van der Waal forces, guys. The van der Waal forces, they are very, very weak. Okay, where they are non-specific interactions. They occur between all atoms, right? So, hmm. So they are basically helping in the close packing of the atoms. That's all. And we have the disulfide bridges, guys. Disulfide are basically the covalent bonds, guys. They are covalent bond formed by the sharing of electrons, and they're formed between the sulfur atoms. They're formed between the sulfur atoms of cysteine residues. Okay, cysteine residue, the sulfur containing amino acids, right? So, which is very, very, these sulfur containing amino acids are basically very, very strong as compared to the non-covalent interactions. So we have these various bonds, which you can just see from here, the peptide, disulfide, non. We have already discussed all this, right? So this is a nutshell, which you can just take away from this particular slide, which is given in the Auricle notes as well, right? What are the fibrous and globular proteins? The fibrous and globular proteins. Now, fibrous are basically, just try to remember, they are elongated. They are rod-like structures. Rod-like structures. Basically, they tend to form chains or fibers, and they are insoluble in water. They are insoluble in water. They are insoluble in water. Basically, what is the function of this fibrous proteins? They actually provide structural support. They provide structural support. For example, you already know we have something called as collagen. Collagen is also a protein. So it is a protein which is found in connective tissues. Okay. Connective tissues, for example, you already know tendon, which helps in the connection of muscle to bone. It is also found in ligaments. It is also found in skin. Okay, so this collagen, it is very, very hard. It provides tensile strength, guys. It provides the tensile strength. Okay, again, one more example that you can remember, keratin. It is found in hair, nails, right? Outer layer of skin. So keratin basically helps in the protection part. Okay, it is like all the bacteria and everything. We have something called as elastin also, right? Now, elastin is basically a protein. For example, if you stretch a rubber band, the rubber band can regain its original shape and size. Similarly, the elastin is a protein that allows the tissue to gain its original shape after stretching or contracting. So this elastin is basically found in lungs. It is found in trachea also, or arteries. Arteries also, obviously. When the left ventricle pumps the blood to the aorta, obviously the aorta is an elastic artery. It expands, and As soon as it pumps the blood to the entire body, it again regains its normal shape. So this elastin is also present in the lungs and also in the walls of the arteries, right? Coming on to the globular proteins, guys, the globular proteins are basically typically like they are spherical. They are spherical, they are oval in shape, right? They are more compact than fibrous protein. They are more compact than fibrous proteins, right? They are soluble in water. They are soluble in water or any biological fluid. So basically, they help in metabolism part, guys. So they help in metabolism, regulation, transport, okay, and various catalysis reaction also. Examples, guys, you already know hemoglobin, myoglobin, myoglobin, right? So hemoglobin, it is basically a protein found in the red blood cells that transport oxygen from the lungs to the tissues. Myoglobin, also you know, it's a muscle protein that stores oxygen and releases when no there is intensive muscular contraction during exercise and all that, right? So you just have to go through this, right? Even the enzymes like lactase, right? So they catalyze the biochemical reaction. They help in the breakdown of this lactose, right? Now, coming on to what is the significance of protein misfolding and its relation to disease like Alzheimer's or prion disease. So, guys, remember one of the things that there is something called as loss of function. Now, when I say loss of function, proper protein folding is very, very important for biological activity. Misfolded proteins are the proteins which are not folded according to the right sequence. They will be like there will be delayed cellular responses, disruption in the cell response. Okay, there could be if the protein is not useful, and this will be something referred to as toxic. So there will be toxic aggregates, right? And yes. So for examples, Alzheimer's disease, guys. The Alzheimer's disease, you already know it is associated with two proteins, beta amyloid, and we have something called as tau protein. They are very, very important and they are often asked in the exam. Beta amyloid and tau protein. The beta amyloid basically it misfolds and aggregates to form plaques. Okay, outside the neurons. Outside the neurons, they will form plaques, right? And it will lead to inflammation and cell damage of the neurons. Tau, when starts misfolding, guys, it basically forms neurofibrillary tangles, right, inside the neurons. So basically, inside the neurons, it will no degenerate it. Okay, and that is something you have to remember. In prion disease also, or prion disease is also called as Creutzfeldt-Jakob disease. Creutzfeldt-Jakob disease. Now, you already know it is caused by misfolded proteins, right? PRPSC, that converts a normal protein into misfolded form. So it can also lead to some neurological symptoms, and basically it is associated with the protein misfolding only. So that was regarding the today's session, guys. So we have discussed the various structure of the proteins, their importance, their stability, and everything. So yes, so I hope you understood, and these are the various social media handles. You can just go on for the update of the next classes. Thank you so much.